The prophase of the first meiotic division is highly modified because it is during this phase that the exchange between homologous chromosomes occurs. At the beginning, each chromosome becomes somewhat condensed and exists in the form of two sister chromatids. The chromatin threads are relatively loose and are not as clearly visible as they are during mitosis. Outside the nucleus, the centrioles migrate to the opposite poles of the cell, giving rise to the meiotic spindle, which is made up of microtubules. However, before the nuclear envelope disappears, the homologous chromosomes recognize each other and bind tightly together. As a result, fragments of homologous chromosomes can be exchanged in a process called crossing over. Afterwards, the chromosomes become thicker and the nuclear envelope disappears. Next, the pairs of homologous chromosomes take up a position on the equator of the cell. This indicates that the cell is in the metaphase of the first meiotic division. Anaphase begins when the fibers of the meiotic spindle cause each chromosome of the homologous pair to move towards the opposite poles of the cell. At this point, the homologous chromosomes become segregated. This process reduces the number of chromosomes in the daughter cells by half. When the chromosomes reach the spindle poles, a short telophase begins. The nuclear envelope is restored and surrounds the daughter nuclei. The spindle disintegrates and cytokinesis starts. The two daughter cells, each containing a reduced number of chromosomes, are the result of the first meiotic division. Now the chromatin begins to loosen somewhat, and a brief interphase begins. Soon afterwards, the daughter cells pass into the prophase of the second meiotic division. The main purpose of this is to segregate the chromosome chromatids into the daughter cells. The second meiotic division resembles mitosis. In prophase, densely packed chromosomes are formed. They are positioned at the equator of the cell at metaphase. In anaphase, they are divided even further, and single chromatids are segregated into the opposite poles of the daughter cells. Each chromatid becomes an independent chromosome. Finally, in telophase, 4 daughter cells are formed. Meiosis results in 4 daughter cells with half the number of chromosomes, each consisting of 1 chromatid.